Hey everybody, it's Mr. Matthew here to talk about how to set up the Term 2 project for algae growth. So in this video, I want to sit down and talk about the materials that go into it. I'm also going to talk about the controls that you will use. I'll also talk a little bit about how you go about doing your measurement using the McFarland Standards. I will not tell you exactly how to go about setting up this lab and how to run it, because that's actually part of the focus of the Term 2 project, is to give you a little bit of variability so that you will play around with these materials methods and figure out how to collect data when given some key variables that you know about. All right, so here we go. All right, so for materials, what you're going to want to use is we're going to use some algae. And we'll have a stock available of algae found in every single classroom, and it basically is going to look like this. It's going to be some sort of container that is going to have a set amount of algae in it that you'll want to draw from. It's useful to have a stock that you guys as a group pull aside and then use as your stock algae, but you can draw back from the classroom stock that we're going to keep available. We have a few large jugs of it. We also have some separate bottles of them. So you'll draw from the classroom stock of algae. You probably will have to decide how much you want to use as a starting amount and keep that consistent through all of your different setups. Now, another thing that you're going to want to choose is your fertilizer. Now, we have some nitrogen fertilizer. We have some phosphorus or P fertilizer. We also have some potassium fertilizer. And these are just the nitrogen, just the phosphorus, just the potassium that you can use. Or we have combined fertilizers that have NP and K. So for example, I know in our classroom we have a 10-10-10 NPK fertilizer. You will want to choose some fertilizer to use, and you'll also want to talk about your concentration of fertilizer that you're going to want to use. You'll want to pick something and then use that consistently throughout your experiment. This is one of those variables that you as a group are going to want to choose to have throughout your experiment. Now, we will also have 50 ml Falcon tubes. Now, a Falcon tube, as I showed you with the algae, is just a conical centrifuge tube. Conical means that it comes down to a point on the bottom. It also has a screw cap on the top, and we have lots of them available. I opened up a whole giant container of them, and I will make 4 50 ml conicals available to every group that is using this, along with a rack so that you'll be able to store your group's work throughout this experiment. And then the last thing you're going to want to use are McFarland Standards. And McFarland Standards we'll talk about in greater detail, but we will have a setup of McFarland Standards at the 0.5, the 1, the 2, the 3, and the 4 concentration, and we'll have them in each of the 3 honors biology rooms that we use during the year. Now, how do I set up my experiment? Now, you're going to want to have some controls. So it's important to note that a control is different than something that is a constant in your experiment. So for example, you're going to want to have all of your tubes exposed to roughly the same amount of light all of the time through the experiment. That's a constant. You're going to want the experiment to take place in the same temperature, and that's going to be a constant. You're going to have roughly the same volume, and actually ideally the exact same amount of volume in every single one of your tubes. That will be a constant. But in terms of controls are things that allow you to determine the validity of the materials and the methods that you're using. So one such negative control that you can use, and this is one that determines sort of what happens to algae over time, is you will take a certain concentration of algae, whatever concentration you've decided to use in your experiment, and you will add that into a tube plus water that will be in place of the fertilizer variable that you'll be adding into your other tubes. A second negative control will be to see what happens to fertilizer over time. So instead of adding algae to your tube, you will add water up to a certain volume and then the appropriate concentration of a volume of the fertilizer that you're going to have. So one of the things that you'll note is that when I have a tube like this and I'm adding a certain amount of fertilizer, I want to not add like 20 mils of fertilizer and 5 mils of algae. I also don't want to add 20 mils of algae and 5 mils of fertilizer. I'm probably going to decide on some sort of concentration volume where maybe I'm adding an initial amount of, let's say, 5 mils of fertilizer and I'm going to add 5 mils of my algae, and then the remaining 25 mils, or whatever constant you've decided in your group, will be of a certain amount of water that you add to that. If you keep that in mind, you could use for these two negative controls, you'll have that algae amount that you're adding to all the tubes, and the rest up to the concentration or the total volume will be water. Or the fertilizer amount that you add to the tubes that have fertilizer, plus the rest of them up with water rather than a mixture of algae and water. And these are two negative controls, and this allows you to see what happens to the visibility or turbidity of the tubes that have just algae in them or just fertilizer in them. Does the algae grow on its own if you don't add fertilizer? Well, the answer is possibly. How are you going to know how much that is? Well, you set up this tube in order to determine it. Does the concentration of fertilizer change over time, or does the turbidity of something that has a fertilizer in it change over time? We don't know. And so a control will allow us to account for any changes that will happen to the fertilizer concentration over time. Now, the positive control is one where you're going to set up a tube where you're going to have a set amount of fertilizer that you think is going to have an impact on this algae growth. Ideally, it's one that will help the algae grow over time. And this will be your positive control. You're going to have some discussions as a group about how much fertilizer you want to add. Obviously, if you add not enough fertilizer to this, you may not get a measurable growth that's much different than, say, your negative control. If you add too much, you might actually burn out or kill your algae. So you don't want to add too much or too high a concentration, because that would have a deleterious effect on your population growth over time. And so you're going to have to discuss and figure out how much you want to add. And this will depend also if you're using a nitrogen fertilizer, a phosphorus fertilizer, a potassium fertilizer, or some combination of the three. And then for your experimental tube, you're going to pick some different amount of fertilizer. So let's say you do your research and you find what your positive control is going to be, and then you want to add double that amount of fertilizer. Is that going to be beneficial to the algae? Is it going to be harmful to the algae? Maybe you want to use 50%. Maybe you want to use 20% more. You'll have to decide this as a group, but you're going to have something that's going to be a different amount of fertilizer from what you set up as your positive control to ask a question, or more importantly, to answer the question you've decided to ask about fertilizer's impact on algae growth, and maybe some parallel to the concept of eutrophication, which is ultimately what we're looking to test in this system. So how are you going to actually measure your dependent variable? So what we end up using is what's known as McFarland Standards. And McFarland Standards are a turbidity standard, which is a stand-in for the amount of cell growth you have. And so what we will have is we will have these standards set up. And so I have everything from a 0.5 McFarland standard up to a 4 McFarland standard. And what these are is there are solutions that are cloudy or turbid. And what we'll use is these are a stand-in for how many cells would grow in the container. And so what I like to use these for is they give us a relative comparison. So when you have a certain amount of algae that's growing in a tube, you can hold this up next to your McFarland Standards. And then what I like to do is I try to read something, usually some typed text, on a piece of paper, and I look at a typed text and I look at my sample of algae, and I hold up next to that the different McFarland Standards. And what I'll find is that some of the McFarland Standards will be roughly the same amount of cloudiness or turbidity as the sample that I'm looking at. Now, in reality, we will only have 5 McFarland Standards, a 0.5, a 1, a 2, a 3, and a 4. So is it possible that what you'll end up having is something that's somewhere between 1 and 2? And the answer is absolutely, in which case you will make a decision. Is it really close to 1, or it's basically the same as 1? Is it really close to 2, or is it somewhere in between? If it's somewhere in between, I would call that a 1.5. Now, what you can use the McFarland Standards for is this gives you a rough estimate of approximately how many cells you have in every milliliter of solution. This does assume that the things that we're looking at are bacterial cells, and so that's actually what we're looking this based off of. And I realize that some algae are actually going to be eukaryotic, and so this is an imperfect measurement, but we're going to use this as our assumption that these are photosynthetic algae and that they may be single cellular. So let's say, for example, we go back to that example I gave you, that 1.5 McFarland standard. It turns out that the approximate number of cells that you have, if you have a McFarland standard of 1, is 3 times 10 to the 8th, or 30 million bacterial cells. And if I had 2 McFarland standard, I would have 60 million cells that I have. And so if I have something that's 1.5, that would be roughly 45 million cells. Now, this will allow me to see at what great growth rate I have in my cells. So if I start with my initial population being something that is below that 0.5 McFarland standard, and now after 5 days, I've moved up to a McFarland standard between 1 and 2, or a 1.5, that can tell me that I've had my population maybe triple in the amount of time I've been running this experiment. That gives me something to tell me about the impact of certain types of fertilizer on population growth. And moreover, I can compare with both my negative controls and positive controls to see how that impact is relative to other conditions. All right, so I hope this is a useful standard for you in order to figure out the rate of algae growth in your different standards. And this gives you some ideas about how to go about collecting data if this is the experimental setup you're going to use. All right, please ask me or your teachers about questions that you have as you're setting this up, and I will talk to you soon.